Welcome to Brain Bites, the bite-sized neuroscience podcast aimed to inform you about the neuroscience world in all short, digestible episodes. This episode is brought to you by the Ascend program, advancing STEM community engagement through neuroscience discovery. I'm your host, Dr. Dan Nemeth. I'm a postdoctoral fellow studying neuroimmunology. And today we have a very special guest with us. We have Professor John F. Cryan. Professor Cryan is not only a leading expert in the field of neuroscience, but he also wears many hats. He is the professor and chair of the Department of Anatomy and Neuroscience at University College Cork in Ireland, and also the vice president of research and innovation. And he's also the principal investigator of the APC Microbiome Institute. He is the co-author of a best-selling book, The Psychobiotic Revolution, Mood, Food, and the New Science of the Gut-Brain Connection. And today he joins us to share his experience on the intriguing relationship between the brain, gut, microbiome, and how it influences our mental health. So welcome here.
Welcome, Dan. Thank you.
So can you tell us a little bit about your background and how you got from, where you started your studies and how you made your way all the way up to studying the microbiome?
Yeah, no, it's been an interesting journey. So I'm Irish, so I grew up on a farm in the west of Ireland, first of my family to go to university, and I studied science, a general science degree that led to majoring in biochemistry. And from there, I was interested in how we could then exploit these cells or, really work on these cells in different ways with certain drugs. So I did my graduate work in pharmacology. I then did part of my PhD in Australia. I then went to the US, to Philadelphia, and after some years there, I decided, you need to do East Coast and West Coast in the US. So I went west to La Jolla, to California, and worked on the relationship between mood disorders and drug addiction. Then I decided that it would be worthwhile going to industry, and I got offered a role as a group leader in the pharmaceutical industry, and that was back in Europe, in Switzerland. Eventually, then I got the homing gene activated, and I decided to return to Ireland. And a new pharmacy school was opening up in Cork, in the very south of Ireland, and so this was kind of appealing to me. And I started collaborating with people who were interested in this thing called the microbiome.
Could you elaborate more on how our gut microbiome or our microbiome is composed, or what is it made of, and then also, like I said, where you can find it, and then also what are the ways in which it can lead to or communicate with the brain?
Yeah, great questions there, Dan. I guess the first thing to say is that the microbiome is made up of the bacteria, the viruses, the fungi, the archaea, and many other single-cellular organisms that we have in our gut. Predominantly is the biggest place, but we have them all in every orifice we have in our body, in our oral cavity, throughout everywhere you can imagine. There are microbes there. I guess it's important to step back a little bit and realize that the microbes were there first. Mm-hmm. We should also remind things also that microbes have antecedents in all cellular activity. In relation to that, you can imagine the mitochondria, which is the powerhouse of every cell, are just microbes that got lost overall. So we've co-evolved with these microbes, and so and we get our bacteria as kind of a birthday present on the day we're born, as we emerge from our mothers, passed on from mum to infant. And these early bacteria then inform the developing immune system and help program many systems in the body overall. So I guess what has become clear is the factors that influence the composition of these microbes in early life could have long-term effects on the way laying down of these frontier bacteria are. So we and others have been studying this, and in both animal models and in humans, and so we know things like mode of nutritional provision. So if an infant is breastfed, it has a very different microbiome than an infant that is bottle fed. One of the reasons is that human breast milk contains the most complexity of sugars than any other animal. So compared to a cow, the sugars are about 20 times more complex in humans.
Interesting. Okay.
And these sugars cannot be broken down by the infant. Now, evolution doesn't do that without a reason. And what happens is they're actually broken down by the microbes. So that's why no matter what we try and do in infant formula, we can never really recapitulate the true beneficial effects of human breast milk because of the complexity of what's going on overall. So that's diet is really important in early life. We do know that exposure to antibiotics or other medications in early life is important. We know that the environment you grow up in, whether it's in a green environment or a city environment where you're exposed to lots of pollutants and various things, will impact it. We've been interested in a mode of delivery at birth because if the microbes are being handed off from mom to infant, but in C-section the baton gets dropped. And so we're not able to make that continuation. And we know that infants born by C-section have a greater propensity for allergy and asthma and type 1 diabetes. We know less about it in terms of brain health, and that's something that we've been studying.
Is that, during our developmental windows, right, during our development, when our brain is starting to form, the lack or the change in bacteria, you're saying that the lack of change of bacteria that we have during those times can actually influence how your neurons might be able to form and function?
Yeah, so that's exactly the point, Dan, is that it's a very dynamic, during early life, there's a lot going on in brain development. And what we're trying to understand now is there's also a lot going on in microbiome development. And then how do they talk to each other? And so if we mess something up on one, can we also mess it up on the other? And I think that's going to be a crucial area for research, and also will give us insight into certain brain disorders as well that emerge early in life that may be due to manipulations of the microbiome.
And could you talk a little bit about, we just went to your lecture and you were talking a little bit about how you are able to kind of replicate the loss of bacteria in these mice and try to understand how that might alter brain development or behaviors later in life. Could you elaborate a little bit on that?
Yeah. So to get evidence that something is important in a process, one of the best ways we have when we look to engineering or other areas is just knock it out and see what happens. And so that's what it means by growing up germ-free. And so we're fortunate enough in our facility in Cork, we have a germ-free unit which allows animals to grow up completely germ-free, never ever having access to microbes at all. They're essentially grow up in a bubble.
Yeah, and they're little bubble boys, little bubble mice.
They're little bubble mice, and we and other groups have shown that these mice, if they grow up like that, their brains don't develop properly.
Could you explain a little bit how we test how mice act in that way?
Sure. We find very distinct changes in their behavior related to stress and social behavior. They have an exaggerated response to an acute stress. So we can measure hormones that are increased by stress called like corticosterone, and that's elevated in these mice. But on a behavior level, we find that they have changes in their ability to remember an association between a fearful situation and a tone. But one of the biggest findings we found was that mice, like humans, are quite social. So if you give them the chance to spend time with another mouse or an object, they'll gravitate towards the other mouse, but not if they're germ-free. So if you give them the chance to spend time with their everyday mate versus a new playmate, they'll gravitate towards a new playmate, but again, not if they're germ-free. That for us was kind of we found this a decade ago, and it was like a real eureka moment of like, oh my God, like this is really intriguing. And so then we started to survey across the entire animal kingdom, and when you look at it, whether you're a bumblebee in a hive or a baboon in the wild, if you interfere with social hierarchies and social dynamics, it's changes in the microbiome you see time and time again, and vice versa.
How do we modify them? Right? Is it the way that we eat, the way that we behave, the way that the people around us like how is the gut microbiome, changing with us as we live?
All of that, Dan. All of that is key. I guess diet is one of the biggest factors, and we know there are certain diets that are good for our microbiome. So increasing fiber and green vegetables and color, and it was really good for our microbiome. Whereas ultra processed foods, sweeteners, have negative impact on the microbiome.
Yeah. So I guess then what would you recommend then, I guess, for a child growing up, like what would be something that would definitely enhance your gut microbiome in a way that is beneficial for us? Yeah,
and it's one of the things that I'm thinking a lot of because my own kids are in this. Mm-hmm. Eat more fiber, but eat more vegetables. These are key things that we can say. I'm a big fan of fermented foods. Mm-hmm. But it's really about diversity and trying to rule out processed food or ultra processed food.
Is there any evidence to show that the way that your neurons are working or reacting to the environment around you actually affects your gut microbiome, and that causes this bad feed forward loop downway into a bad behavior?
That's an excellent question, Dan. I mean, I think there are. The best data we have is on stress itself, which is kind of top down. There is also some really intriguing data on brain injury, so that TBI, which is a direct effect on the brain, is also changing both microbiome and gut inflammation. Mm-hmm. So that there is a top down. I think we need more studies in humans. It would be really nice to see things like yoga or mindfulness or various other meditation practices, whether they could have a positive effect on the microbiome in that way.
What do you think was one of the most exciting discoveries that you've made in your course of your research? I
guess one of the most important discoveries for us was when we showed that in mice that grew up without microbes, and we did this completely unbiased. We looked at all the different genes that are changed in an area of the brain that's involved in higher cognitive functions, so the prefrontal cortex, and what we found was when we looked at these genes, there was an upregulation, so there was more of genes that were involved in the process known as myelination. Now, myelin is, as you know, Dan, but just for your listeners, is an important insulation on brain cells. It allows them, basically like you have insulation in your home, myelin is insulation on your nerve cells to actually allow them to conduct appropriately. And we mainly think about myelin in the context of demyelinating disorders like multiple sclerosis, which cause when your immune cells are attacking the myelin. Here we got an upregulation. So that was really striking for me, but because it meant, conceptually, could we now target myelin process in the brain through the gut? The second one was kind of a bizarre study we did to prove if we think that the microbiome is important for brain aging, then we should be able to prove that by taking the microbes from young animals and giving it to older animals, we should be able to slow down the effects of aging. And we did that. And, it was really remarkable. We took poop from young animals at 8 weeks old, gave it to 22-month-old animals, and they're completely rejuvenated their brain.
Oh, wow.
And their behavior. And so nowadays people are looking at new ways of delivering this medicine in different ways. So, for example, they are able to put it into a kind of a tablet or a tablet, and people are testing these types of approaches in depression, bipolar, and other illnesses right now.
Is there any upcoming projects that you're super excited about that you're chomping at the bit to get finished?
Yeah, we have one project that it's on the relationship between our circadian biology and the microbiome. And it's really intriguing because almost all systems in our body change throughout the day. Mm-hmm. And a little known fact is that your microbes change
Oh, wow.
throughout the day. So when you're like me now, who came from Europe yesterday, so when I got in last night, my microbes were like, "What the hell is going on here?" He's not eating at this time of night. You're not supposed to get food at this time of night. So we're beginning to realize it's not what you eat as much as when you eat, at least for your microbes overall. But we've shown in these studies that there seems to be a correlation between how stress hormones function throughout the day and the microbiome, and we're trying to look at this causally overall. And because circadian rhythms are such an important part of mental health, and in many mental health disorders is disturbance of your sleep and circadian patterns. So the question I have now is, can I harness the power of the microbiome to look at this in different ways?
I guess, we're going to wrap up shortly here. We're going to do like a quick rapid fire type question type thing. So what's your favorite, brain boosting snack?
Probably not the tastiest, but for the best for your microbiome, I think will be you're having, for us, it's a fermented food called kefir.
And besides your own book, what do you think one book you think that budding neuroscientists or budding scientists in general should read?
I mean, one that I liked at the time was Apprentice to Genius,
okay.
Which was about the politics of lab work. That was really important overall. I just got a present of Cahal's Advice for a Young Investigator, which is also a good read. And I guess Descartes' Error by Damasio is also a really good. But these maybe for more slightly older.
Okay. Once you get into college level.
Yeah.
And then for our young students who might be interested in pursuing a STEM career, do you have any advice for them going forward?
Yeah, especially to young girls, please do not just leave it to the boys to try and solve problems that are going to affect you. So please, we need more women in science. We need more women staying in science, and we need more creative minds who are interested in bridging disciplines in science.
Oh, definitely. Awesome. Well, thank you so much for your time and for all the insight you have about our gut microbiome and how it affects our brain. And I was really excited to get to talk to you today. And so hopefully everyone will enjoy it. And I'm excited to put this out and have our students listen.
Thank you, Dan. My pleasure.
Thank you so much.
No problem.
Okay, awesome. So I'm Dr. Nemeth, signing off till next time. Brain Bites is directed and produced by Danny Nemeth. Music's by Max Hentosh. All of this is made possible by the generosity of the Stiles Nicholson Foundation, in coordination with Florida Atlantic University, the Max Planck Florida Institute, and the UF Scripps Research Institute of Florida. Thanks. See you next time.